Hey everyone, welcome back to my podcast, Anatomy and Physiology Bit by Bit. This is your host, Dr. Steve Sullivan, coming to you from Bucks County, Pennsylvania, where I've been teaching anatomy and physiology at Bucks County Community College since 2002. Well, I'm glad to have you back with me. I'm trying to get through the heart today. I'm going to do some talk about the function of cardiac muscle tissue in the heart. So we'll talk about the structure of cardiac muscle cells, and we'll talk about how the heart muscle actually generates its own electrical signals. So the heart's really interesting because it doesn't require a nervous innervation to be stimulated to contract. It actually generates its own electrical signals, and we call that the autorhythmicity of cardiac muscle cells. So we'll talk about that as well. But before we get into that, I want to talk about something that I got into a little bit at the end of the last episode when we talked about the coronary circulation. When you consider the arteries that are supplying blood to the heart muscle itself, it is really, really important that those arteries stay open and the blood can flow through easily and they do not narrow so much that a blood clot can actually block the blood flow getting to the heart muscle. And that is the problem. Coronary artery disease, the #1 cause of death in the world, can be caused by lots of different things. Genetics, for one, which is not something you can really do a lot about. They call that a non-modifiable risk factor, but there are also modifiable risk factors, like leading a sedentary lifestyle where you don't get enough exercise, eating diets high in saturated fats and sugars, being overweight, smoking, right? These are some of the things that will cause coronary artery disease and make it so that these arteries can't get the blood to where they need to go. When those blockages become really severe, there might need to be a surgical intervention to restore blood flow to the region of the heart. And one of the main versions of those is a coronary bypass operation. So you've probably heard of that. You may have heard of double bypass, triple bypass, quadruple bypass. So basically what they do is they find the occluded artery and they take a vein from the thigh. They go in surgically, cut the artery before the blockage, attach the vein to it, and then run the vein to the artery after the blockage and reattach it in there so that the blood actually bypasses the blockage. And if they do that for one artery, it's a coronary bypass. If they need to do 2, it's a double. 3 is a triple. 4 is a quadruple. What they do is they try to make sure that the blood can still get to the heart tissue the way it's supposed to, and that it's not going through a vessel that's become so occluded or stenotic that a regular-sized blood clot will block the blood flow to the area. When you block the blood flow to an area, it's called ischemia. That leads to an oxygen deprivation in the tissue, which is called hypoxia, and then that can lead to an infarct, which is a death of the tissue. And if that tissue is the myocardium, the heart muscle, it's a myocardial infarction, or MI, which we commonly know as a heart attack. So sometimes that coronary artery disease, the occluded vessels, will cause symptoms like pain and pressure in the heart. And we call that angina, which can sometimes feel like heartburn or a muscle strain in the pecs. So I just wanted to touch on that clinical aspect of the previous episode that we didn't get into because it is so significant to any healthcare practitioner. So having said that, let's move on with the heart, and we'll begin with talking about cardiac muscle histology. Cardiac muscle tissue is unique when you compare it to the other kinds of muscle tissue like smooth muscle and skeletal muscle. And understanding what a cardiac muscle cell is and the anatomy of it and how it's connected to other cardiac muscle cells is really important to making sure that you can understand the function of cardiac muscle tissue. So we're going to get into that first. We'll talk about the histology of cardiac muscle tissue. We'll look at a cardiomyocyte, which is a muscle cell in the heart. And then we'll have time to see what the function is based on that structure. Like skeletal muscle, cardiac muscle is striated, so it looks kind of striped with overlapping thick and thin filaments, which are made mostly of the same proteins that we had in skeletal muscle, myosin for the thick filaments and actin for the thin filaments. The difference is that instead of being long, thin, straight muscle fibers like we had with skeletal muscle, the cardiomyocytes are shorter, they're thick, and they actually are branched, like they fork. Cardiomyocytes can have one or more nuclei, so they can be multinucleated, like some other cells, and each of them are surrounded by a mass of glycogen. Glycogen is the storage form of glucose, and since our heart muscle has to contract over 100,000 times a day, it needs a ready supply of energy, and it does that in the storage form of glucose called glycogen. Cardiomyocytes have a sarcoplasmic reticulum, and they have transverse tubules, but they don't have any terminal cisterns. So again, the difference between them and skeletal muscle cells. Calcium ions from the extracellular fluid can enter the cell via those transverse tubules, and then they can use their charge of 2+ to help execute the muscle contraction required in cardiomyocytes. Another real unique thing about cardiomyocytes is that they conduct their own electrical signal and they transfer that electrical signal to adjacent cardiomyocytes. They do this because they are bound to one another with gap junctions. Now, the boundary between one cardiomyocyte and another is called an intercalated disc. And in those intercalated discs are these gap junctions. In fact, the word intercalated means inserted between two others. The gap junctions are going to allow the ions in the cytosol to flow directly from one cell to the adjacent cell. And this way, we don't have to have separate neurons innervating different cardiomyocytes trying to coordinate this wave-like contraction we need. Instead, the depolarization of the membrane flows from cell to cell through these gap junctions. The sarcolemma of each cardiomyocyte is folded into finger-like projections called interdigitating folds. And what they can do is those interdigitating folds interlock with one another, connecting the two adjacent cardiomyocytes and maximizing the surface area that are linking those two cells together. Now remember, sarcolemma is the word we use for the plasma membrane of a muscle cell. Throughout those connected interdigitating folds, the actin of the thin filaments extends and attaches to the cytoplasmic side of the plasma membrane. Transmembrane proteins of adjacent cardiomyocytes connect the cells to one another, and that structure is called the fascia adherens. Now, in addition to the gap junctions, those fascia adherens have desmosomes, and those are strong, protein-based cell junctions that resist the tensile forces of contracting cardiac muscle. So when a cardiac muscle contracts, a heart is beating and it is pumping blood, and it's very strong. And what you don't want is the cells to be torn apart from that force. So these desmosomes can resist the separating of the cardiac muscle cells from all that force. The last component of the intercalated disc is those gap junctions. Gap junctions function as tunnels between adjacent cells that can allow cytosol and small solutes to pass directly from one cell to another. The transmembrane protein of a gap junction is called a connexon. It goes from one cell's plasma membrane, or sarcolemma, to attach to an adjacent cell's sarcolemma. Like I said before, in cardiac muscle cells, ions can flow from one cell to the next through these gap junctions, resulting in an organized depolarization of the sarcolemma. This allows the cardiomyocytes of the heart's chamber to contract in unison without the need for each muscle cell to be innervated by its own motor neuron. Now in skeletal muscle, that's what we had. We had these motor units where you had a skeletal muscle like the biceps brachii, and there are several motor neurons leading to the different muscle cells of that whole muscle. And the number of neurons that you engage when you go to contract your biceps brachii will determine how much strength you will use to contract your biceps brachii. Maybe you only want to bend your elbows 90 degrees. Maybe you're only lifting a coffee mug rather than a bowling ball. So you need to recruit more muscle cells for a bowling ball than for a coffee mug. And we called that recruitment. So those skeletal muscles had these motor units, these regions of muscle fibers that are all innervated by the same motor neuron. We don't have that in cardiac muscle. In cardiac muscle, you've got cells that are generating their own electrical signals without the need to be innervated by a motor neuron, and then they transfer that depolarization, that excitement of the muscle cell, to the adjacent muscle cells through these gap junctions. If we were to use a light microscope, we could see the overlapped plasma membrane of the intercalated discs, and it shows as darkly stained bands between each cardiomyocyte. Now, like I said before, the typical adult heart contracts about 100,000 times a day. So it needs a lot of energy to do all that work. And in addition to the glycogen stores, cardiac muscle tissue also has myoglobin. Myoglobin is a protein that binds oxygen for use when it's needed. It also has an ample blood supply, lots of mitochondria, and creatine kinase, which contributes a phosphate group to adenosine diphosphate, resulting in adenosine triphosphate. About 25% of the volume of a myocardiocyte is made up of mitochondria. Now, the reason for this is because we need a lot of ATP to sustain all of this work. And that ATP is mostly coming by way of aerobic respiration, which takes place in mitochondria. Cardiac muscle is really good at utilizing different substrates as energy sources. Glucose, fatty acids, amino acids, lactate, and ketone bodies all can be utilized by cardiomyocytes. Remember, you've got something so important, something that's pumping blood to your brain, and it needs to do a lot of work and it needs a lot of energy. So it's going to take whatever it can get. It's going to store glucose for itself. It's going to store oxygen for itself in myoglobin. It's going to be ready to go all the time. Since aerobic respiration is essentially the sole mechanism for ATP production in cardiac muscle, a lack of oxygen is extremely dangerous. So like I opened up this episode talking about the coronary artery disease. When you narrow a vessel, it's called stenosis. And if a coronary artery becomes stenotic, the blood flow is going to be impeded to the muscle tissue, and that is no good. Like I mentioned earlier, we know what can happen with that, and no surprise, it is the #1 cause of death in the entire world because of how dangerous that is. Okay, so now that we have an idea of the histology of these cardiomyocytes, I think it's really important to talk about the autorhythmicity of cardiomyocytes, how they can generate their own electrical signals. Now, autorhythmicity means it makes its own rhythm, right? Like an autobiography is written by the person who the book is about. Auto means self. And the specific autorhythmicity that we're talking about are in a particular part of the right atrium of the heart. And that area is called the sinoatrial node. Sometimes we say SA node. So we say the sinoatrial nodal cells have the capacity for autorhythmicity. Like any other cells, these cells have a plasma membrane. They also have a membrane potential due to the difference in ions inside and outside of the cell. The resting membrane potential in the nodal cells is minus 60 millivolts. But it's not really a stable resting membrane potential. And that's because there are voltage-gated sodium ion channels that let positively charged sodium ions slowly leak in because they're open when the cell's at rest. These are called slow voltage-gated sodium ion channels. And at the same time, potassium ion channels are closed. so they don't counteract all these positive ions coming in. Remember, typically potassium ions are in higher concentration inside a cell than outside a cell. So if they were open too, we'd have potassium ions leaving the cell at the same time, and they're also positively charged. So we would counteract the changes in membrane potential created by these slow sodium ion channels. So since those nodal cells' membrane potential is slowly becoming less negative without an outside stimulus, we're going to have the potential for them to create their own local potentials without having any kind of outside stimulus. In addition to those sodium ion channels, the SA node also has voltage-gated calcium ion channels. Remember, calcium ions have a 2+ charge. The voltage that stimulates the opening of its gates is minus 40 millivolts, and we'll call that threshold. When the nodal cell's membrane potential reaches that threshold, it opens, and calcium ions, which are in higher concentration outside the cell, rush in very quickly. They will go down their concentration gradient, and because it comes in so fast, we call those fast voltage-gated calcium ion channels. The influx of those cations with a charge of +2 causes a rapid depolarization that reaches just above 0 millivolts. At that point, the calcium ion channels close and the influx of calcium ions stops. This is when the voltage-gated potassium ion channels finally open. And since positively charged potassium ions are in higher concentration inside the cell, they flow out down their concentration gradient and bring the membrane potential back into the negative. In fact, the potassium ion outflow brings it all the way back to minus 60 millivolts, the original resting membrane potential of the nodal cardiomyocytes. We call this the repolarization of the membrane, and it stimulates the opening of those slow voltage-gated sodium ion channels. Remember, that's where we were in the beginning when we started this whole process. Since the SA node sets the pace for your heart, its nickname is the pacemaker of the heart. Under normal circumstances, the rhythm of the heart is called sinus rhythm because the pace is being set by the sinoatrial node. You might have seen this on a TV show like about hospitals and doctors where they are situation in an emergency room, they have someone whose heart is in VFib and they have to bring out the paddles and shock them. And when they do that, once you see the machine start going beep, 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 they'll say, we have normal sinus rhythm. That's what they're talking about. That means that we've got the rhythm coming from the SA node again, like it should be. Now, sometimes these autorhythmic cells, they start to fire off randomly and from different places other than the SA node. When that happens, we call that an ectopic focus, meaning that we have the focus of the beginning of the heartbeat coming from somewhere other than where it's supposed to come from. When that happens, we'll have an abnormal heart rhythm, and we generally call that an arrhythmia. And there's lots of different kinds of arrhythmias, but in general, an arrhythmia is when the heart rhythm is not normal. So when we talk about how to interpret an ECG, an electrocardiogram, we will talk about arrhythmias because we use that machine to interpret what is happening with the electrical activity of the heart. So if there is something going on with the heart that's not right, we can use an ECG to examine the electrical activity of the heart, and then we could see if there's something wrong, if something's not doing what it's supposed to be doing. But that's going to have to wait for another episode because in order to understand an ECG and be able to interpret it correctly, you have to know the cardiac conduction system, which is how the cells of the heart actually conduct the electrical signals generated by the SA node to the rest of the cardiomyocytes so that they can be stimulated to contract. We'll do that in another episode because I think this was enough information for one. Once again, thank you so much for joining me. I really appreciate it. Don't forget that you can always email me at minus55media@gmail.com, and I would be happy to answer your questions. I may even answer them on an episode. I hope this is helping you get your B or better in A&P. Good luck on your next exam, and I'll talk to you next time. Anatomy and Physiology Bit by Bit is a production of Minus 55 Media. please take the time to rate the podcast, and don't forget to check out my YouTube channel, Student Help for AP. That's Student Help, the number 4, AP. There's a whole lot of tutor videos on there that I think you're going to find helpful. Special thanks to my family, Bucks County Community College, and McGraw-Hill Education, where you can find Anatomy and Physiology Digital Suite, my low-cost, tutor video-based digital learning solution for anatomy and physiology, already being used at several colleges and universities.